Good evening, class. Please join in. Join in, students. Good evening, बच्चे. Good evening. I not write the review in two column. No, you don't have to write review in two column patterns. You have to write the assignment in two column pattern. 2 page. So there's a thing called assignment, and there is thing called uh, assignment, and there is thing called review. In the assignment, you have to send me your everyday 2 page summary, and review it could be 7 to 8 page where you have to summarize and one topic that I've suggested and go with that. So there's a difference in between both of them. You meant two column pattern. Sorry, I took you wrong. Sorry, Sagar. Yeah, you can write. You can write. Go ahead. I like that pattern also. Yeah, I understood. You can write. So like you are saying like this, I guess. I had some assignments in that way also. You mean like this in these two column patterns. You can write it. I like this pattern. Go ahead with it. Happy evening, happy evening. So we start in few minutes then. So I hope you all have joined us. It's 8 o'clock. So today I will be dealing with the practical part of yours. Uh, so few practicals that I taught during this course, from chi-square test, colchicine treatment, monohybrid, dihybrid, mitosis, meiosis, all these things we will deal today. So shall I wait for a few minutes to let the people add in, or shall we start? Okay, we should start then. It's already 1 minute left from 2. So want to start from where? Uh, okay. So this chi-square analysis, it is done to check how is your Mendelian method is fit to the model. If 3 is to 1 ratio is coming in a case, how fit is that model is, how much figures are quite true to the 3 is to 1 ratio, how true it is 1 to 1 ratio, 9 is to 1 ratio, how true fit is model. We do this test called as chi-square analysis. So in this case, in this biological experiments, field surveys, apart from quantitative data, we get the qualitative data, which is genetically characters such as tall, short, and color of flowers, seed code, which do not have any numerical values. But the number of flowers or seeds having particular color falls under category can be counted numerically. And this type of observations requires calculation of the expected number of individuals under any category. So thus, it becomes necessary to know whether there is a deviation in between the observed and expected frequencies. The measurement of these deviation is done with the help of particular test. Then we do, which is called as a chi-square test. Then in that case, we make the chi-square test. The formula for chi-square test is uh, addition of observed minus expected whole square divided by expected values. So O is observed value, E is your expected value, and then that's how you calculate your chi-square value. So what are the applications of chi-square test? Is it is an alternative test to find the significance of difference in two or more than proportions. It can compare value of two binomial samples that they are why they are small. It can compare frequencies of two multimonial samples, and chi-square test is applied as a test for goodness of fit, how fit is your test is, which reveals the closeness of the observed frequency with the expected frequency. So goodness of fit, what does it mean? That is applied to wide range of studies related to experimental studies. The aim of this test is to test closeliness of observed samples, how well the observed frequency curve fits into theoretical curve. So if both uh, sorry Just a second. Okay. If both observed and expected frequency distributions are in complete agreement with each other, then chi-square value will be 0. If both what you have observed and what is expected, both values are in agreement with each other, then chi-square will be 0. But in experimental observation, there is always some degree of deviations, but we always see some deviation from the experiments and then real value. So critical chi-square value will be exceeded due to the sampling fluctuations. For example, crossing experiment gives two different sizes of seeds in F2 progeny, and these two types of seeds, according to the Mendelian hybrid, that is, if it is Mendelian monohybrid, then it is 3 is to 1. If it test cross, then 1 is to 1. Complementary factor interaction, then 9 is to 7. Inhibitory factor, 13 is to 3. Duplicate gene interaction, 15 is to 1. So these are the various ratios that we get in our case of um, F2 generations. So we have to now, so how do we do this chi-square test, goodness of fit? We do it by checking the deviation first, by between the observed and expected results should be calculated, then compare the minimum deviation of null hypothesis should be selected of the chi-square test, then chi-square should be determined, and then this value has to be compared with the tabulated value. Conclusion has to be made then later on. So I will show you how it is done. That was done uh, that was done in the other way. Now let's check another way. uh, Following number of seeds with associated character is observed, test the goodness of fit and comment. So this is yellow seed 428 and this is green seed 152. uh, so yellow seed is and green seeds are 152, so we have to calculate the expected value for each ratio. So let's calculate first for total number of samples we have to calculate. So it is 428 plus 152, that is 580. Then expected value can be calculated as for the 3 to 1 ratio to be 3 by 4 multiplied by 580 is equal to the 435. And expected green seeds value will be is 1 ratio 1 by 4 multiplied by 580 is equal to the 145. So these are expected yellow seeds values and these are expected green values. So but in reality the observed values are 428 and 152. So there is deviation of how much of deviation there is? There is deviation of 7. Same way we calculate for the 1 is to 1 ratio expected value for yellow and green seeds by 1 by divided by 2 multiplied by 580 is equal to 290. So in both cases it is 290. Yellow seeds and green seeds equal amount. And 9 is to 7, then we add them. 9 plus 7 is 16. Then 9 by 16, 7 by 16 multiplied by 580 and this way, and we calculate all the different ratios values, then different expected values and different green seeds values. So determination of expected segregation ratio, here you can see uh, there is observed values. So the observed values are yellow is 428 and green is 152, and total deviation could be find out to be here. So expected in the case of 3 is to 1 ratio, expected value was 435 and 145. So how much deviation do we have? We minus from 435 to 428, it comes out to be 7 and also 7 here. So we add them together, total deviation is 14. And same way we'll calculate in other cases the deviations. So these values 138, 138, we are calculating from here, from these values. So these are deviations values are subtracting from the expected values. So and we then adding them, and then we can see that according to the table the minimum ratio could be found in the 3 is to 1 ratio. Minimum value is in the 3 is to 1 ratio, so observed samples should fit 3 is to 1 ratio in this case here. Okay, so following number of seeds with the associated character is observed, Uh, so to calculate now chi-square value for the 428 and 152, observed values are these, expected values are these. Then we do that formula O minus E, then it come out to be O minus 7, and in this case to be plus 7. Then we made a square of it, 49, 49, and then we square is multiplied by expected value, that is 435, and here by 145. And then in the summation, this is addition of these values. We add these values, then values come out to be 0.451. So there are two classes, we can see there are two classes. So degree of freedom formula is 2 minus 1 is 1. So degree of freedom is coming out to be 1. So chi-square value is 0.0451 and degree of freedom coming out to be 1. So falling number of seeds associated in the observed uh, test uh, test. So we have to check that whether it's a goodness of fit or not. So with this case, so we do a conclusion that the chi-square value is 0.451, right? And tabulated chi-square value for degree of one freedom at the rate of 0.05. So how you can calculate this value? Tabulated value is by this one. So at the degree of freedom 1 with 0.05, it's coming out to be 3.84. So this is chi-square distribution table where you can find the degree of freedom here, and this is the level of yeah, some value that you have to check at 0.04 always, and it's coming out to be 3.84. What IPL बोलते हैं? So that value was 3.841, and it is quite less than the expected value, than the tabulated value. So the deviation is insignificant, and the observed deviation is due to the chance factor only. It lies in the probability range of 50 to 70%. So the observed result is a good fit with Mendelian monohybrid 3:1 ratio. So this value of 0.451 in this case of 428 152 is expected to be right value, and this is okay. So it is concluded that the experimental result shows monohybrid ratio uh, a ratio by 3:1 ratio. So this is another example. So the assumed genotype of the parent seems to be like this. So you have yellow, green, selfed. You cross them YY, and and you have YY, then yellow, yellow, green, 3:1 ratio. So this is the genotypic ratio of the same. So I hope this chi-square method is clear to you. That was quite easy and straightforward. You just have to uh, use this formula. First you calculate expected and deviation, then you find the which one is the least value, like in this case 14. Then you take this to the formula for O minus square, O minus E whole square divided by E, by putting them in this table, and then you have to calculate degree of freedom, and then from that chi-square table you calculate the value, and then according to that it's like 3.84, you compare it, and then your result is there. Tada! As simple as that. So I share with you now this chi-square test and this one also now itself. Chi-square, chi-square. This I have also attached the table so that you can use it. So let's go to the next part, that is chromosomal aberrations. So yesterday, as we were talking about chromosomal aberrations, like disturbance in the chromosome number by allouploidy, polyploidy, monosomic, trisomic. So like their patterns were increased, they were also decreased. The number of pairs were increased, the number of pairs were decreased, increase in number of chromosome, decrease in number of chromosomes. So there were many aberrations that we saw in the chromosome yesterday in the case of mutations. So today I'm taking that part in the kind of experiments from this paper by Mirsad Kabravichik. They did one induction of chromosomal aberrations in the allium cepa, that is in onion, by exposure of cells to benzopyrene. They use this chemical benzopyrene, and they try to change its mitosis and meiosis phases in the later case. So let's go through this paper. This we had open already. So I'm discussing this paper with you now in presentation style. Um, So this is the abstract of the paper. So in this case, what they're saying is that higher plants have been proposed as a test organism for detection of genotypic substances in environment, and several plant test systems are already in use to check their sensitivity and they're reliable for the short-term assist test. So Allium cepa is one of these plants which has been used in different studies to detect chromosome aberrations induced by chemicals. So because in allium cepa it is quite easy to see your meiosis happening, so that's why we use this system to check what is happening at the chromosome level. So this is better method because we are not using animals in vitro, and it's non-hazardous, and we can check any chemicals, that is how things are happening in this case, and we can predict those effects in humans also. So in vitro use of principally screening purpose for generating toxicological profiles. Numerous chemicals can generate breakage or interchange of DNA segments between chromosomal structures. So allium test is a screening method for to check the genotoxicity evolution of different chemical substance, including polycyclic aromatic hydrocarbons. So that's representative member is BAP, that is benzopyrene. Uh, so BAP is the one which is a representative this polycyclic aromatic hydrocarbons, and we're checking the genotoxicity of allium router chromosome assay. So the treatment with a different concentration of BAP ranging from 1 to 50 microgram per ml are used respectively. And BAP cause, what does the BAP did in these experiments? It decreased the mitotic index and increased the frequency of abnormal mitosis. And when compared with the control, as compared to control, mitotic index was decreased and increased the frequency of your abnormal mitosis. So you will learn also the how the review looks like, how the paper looks like. So you have to tell each and everything and quite nice summary in that part. So this is your PAU BAP. The structures look like this. It's a 1 2 3 5 benzene ring with a molecular weight of 252.3, and it is characterized insoluble to water and soluble in the insoluble in water and soluble in the organic solvents. So that's how your BAP looks like, benzoperine. And something about BAP. So these are the results of various concentrations. So they used various concentration for different hours. So here you can see that concentration of 1 gram per ml, 10 and 50, 1, 10 and 50. There's a control with DMSO, and the experiments are done with the BAP for 24 hours and 48 hours. So that's how they have planned the experiments pharmacologically. Uh, so here you can see that uh, Mi is your mitotic index, P is your prophase, M is anaphase, and M by P is the metaphases to prophase ratio. So what you can see is, as compared to the control, everything is getting decreased. So you can see metaphase is to prophase ratio. It is decreased as compared to the control. For 24 hours, with increase in concentration, it's keep on decreasing. Even with the hours also, 48 hours also, we are showing a decrease in the metaphases to prophase ratio. So that is a mitotic aberration. So chromosomal distribution is not happening in a right way. And further, so in that case, as I showed you, they use different chemicals for different hours, and they use DMSO also, and for the treatment PAU. So the results, we can see that in the BARB effects on the mitotic index, that BARB concentration usually decreases the mitotic activity in significant values compared to the control values. So it is decreased in that case, as we can see in the table, and highest reduction of MI is established at the concentration of 50 microgram per ml in the value of 7.01, and treatment lasting for 48 hours. So when we use 50 and for 48, we saw the least change in it, in the mitotic activity. It's also affect the mitotic distributions. So we can see how it's changing the chromosomal aberrations also. So it is done at the 200 different cells, and they check various aspects of it. So like they check prolonged prophase, how long it is. They check prometaphase. They check vagrant chromosomes. They check sticky chromosomes as such. They check laggard chromosome. These are all abnormalities in your chromosomes. So, so they also check polyploidy, aberrant cells. So what we can see as compared to control, when a BARP is there, most of the things are increased. It is increasing, and at the 50 and 48 hours, it is the highest increased here. 8, 14.8, 2.8, 3.7, 10.7. So everything, your aberrant cells, your polyploidy, your sticky cells, your laggard chromosomes, vagrant chromosomes, prometaphase, everything is increased. So all the things with this chemical, we saw these changes. And this is the some cell figures we can see. In the A case, we can see that anaphase bridge with lost chromosomes. So there is no anaphase in the bridge. There is, in the B figure, we can see disorder of the chromosome kinetics. Chromosomes are not moving as they should be. In the C case, telophase bridge is there, we can see, and D is the sticky chromosomes. So they become flat like worm. So they are not as the chromosome should be. So they are all separated and act like a sticky chromosomes. So these are some statistical analysis how they did. So in conclusion, Aluma SEPA test is a quick, available, easily performable test. You can use any other chemicals also to check the genotoxicity, and we check in this case environmental pollutant, including PAU. So this is better than using humans. So these are the various results that could be proved, that rather than using human and mouse cells, we can use this allium sepals, their different cells, to check it the chromosomal aberrations, and this could be replicated in the humans also then later on. So that's it for this lecture. This was quite small one, so I don't forget, so I keep sharing with you chromosomal aberrations. This one and this one, full paper. So let's start with the third one. So these are various small practicals that are quite important in principle of genetics. So I thought that they should be also included today. So I'm touching them and going further. So in this chapter, we will have a touch a little bit about how the mutation is done, polyploidy is done, that is increase in number of a chromosome. It could be done by radiations, it could be done by injury. In the case of radiation, polyploidy can be induced in plants by exposing their certain parts, such as vegetative buds, flower buds, to radiation for shorter wavelength, like ultraviolet rays, X-rays, gamma rays. And irradiation increases the rate of cell division, also cause the multiplication of chromosome number. So by injury, when the meristematic zone of a plant are injured, the cells at the point of injury grow rapidly and form a callus. And callus growth is enhanced by a chemical substance named coumarin, which also bring about the somatic doubling of chromosomes. We can also do it by regeneration in vitro. Polyploidy is a common feature in the culture tissue in vitro. Some of the plants regenerated from the callus in suspension culture may be found to be polyploids. Polyploids have been developed from callus cultures of Nicotiana, Datura, rice, several other species. By chemical treatment, a number of chemicals are known which induce polyploid in plants. Some of them are colchicine, hydroquinoline, acetophenone, nitrogen oxides, granosan, chloroform, chloral hydrate, some narcotics, alkaloids, some verathane sulfates, acetylphane. So all these could be used for your chemical treatments. So, but colchicine is the best chemical for this purpose. The formula for the colchicine is C22O6N. The chemical formula for colchicine is C22O6 and N. So colchicine, what is colchicine is? It is a chemical compound which was first discovered by Paris in 1889 and demonstrated first by Levin to be specific and efficient chemical to be treated to do polyploidy restitution at nuclei. So colchicine is obtained from the extract of seeds and corms of Colchicum autumnale, family of Liliaceae. So it is obtained from Colchicum luteum and Glorissa superba. So method of application of colchicine, it is as follows. So colchicine treatment is done in one of the following ways. You do first is seed treatment. Dries or soaked seeds are soaked in the aqueous solution of the colchicine of different strength in shallow container to facilitate aeration. Colchicine treatment is given for definite period which is different for different seeds. And after the seeds are soaked in colchicine solution for desired period, they are washed through in water and then they are sown. And treatment of dry seeds give better results than the soaked seeds in some cases. So we just soak them in colchicine. Then seedling treatment. Seedlings may be treated in young stage. During treatment, the shoot tips are dipped in 0.2% of colchicine solution and root tips are covered with the cotton soaked in water, and treatment will be given 3 to 24 hours. In some cases, treatment has to be repeated for 2 and 3rd day also. And then treatment of growing buds of shoots. In some cases, growing points are treated with 0.1 to 0.5% solution of colchicine, which is applied by the brush or with the dropper. Sometimes cotton wool soaked in the aqueous solution of colchicine is applied over the growing point of plant. And treatment is done once or twice for few days. Alternatively, you can do 0.2 to 0.5% of colchicine solution is mixed with the lanolin paste and smeared on the shoot apex. And this treatment may be treated 2 to 3 times daily for a week. In woody plants, 1% colchicine solutions may be applied over the growing bud for proper penetration of colchicine. Solution is applied 2 to 3 times a day for a week. Effect of colchicine. So when colchicine is applied, some irregularities occur in the mitotic divisions. It has no effect on doubling of chromosomes, which proceeds in normal fashion. This chemical is spindle poison and it simply checks the anaphasic movement. So they are like acting as a spindle poison. So they check the anaphasic movement of the chromosomes in two poles. Consequently, mitosis fails to complete. So mitosis does not take place. So double chromosomes become bonded by nuclear membrane. Thus, the restitution of nucleus with double chromosome is found. So we found double chromosome instead of single chromosomes in this case. So allopolyploidy in crop breeding, it has been used for three purposes: utilization as bridging species and transfer of desirable characters from one species to another, for the production of new crop species, for widening the genetic base of existing alloploid crop species. So these are the various aspects of it. Uh, so I think my reason to show you this thing was mainly to show the how polyploidy of colchicine could affect, and these are the various way of doing it. So it's showing that N sylvestris and N tabacum is hybrid sterile, and we cross artificial doubling of chromosomes with the help of colchicine. And then there is synthetic amphidiploid is made, and then we again made a cross with the tetraploid with the tabacum, and then we had a hybrid back cross with the tabacum here. Then we have a pentaploid progeny with two N chromosomes. This is how you can increase your number of chromosomes. That's is showing us about. So that's it about colchicine, how it can double your chromosomes by acting at the anaphase phase by stopping the mitotic interface in it. So on that note, let's take a 10 minutes break or 5 minutes break now, and then we see us for the next part. We have three left part now. We do it then later after 10 minutes break. Or shall we continue like this? Shall we continue, students, or do you need a break now? Just let me know. What do you say? Thumbs up if you need a break. So we do it accordingly. Okay, then today I think we are fresh. We can continue. Moreover, so polyploidy is done, chromosomal aberration is done, chi-square method is done. So we done three practicals so far. Now comes the fourth part. Uh, that is also comes in your lectures. That is mitosis and meiosis. Mitosis and meiosis. Okay. So I hope you all aware of mitosis and meiosis before. So actively dividing accurate cell passes through a series of stages that is collectively known as cell cycles. So there are two gap phases, G1 and G2, and there is S phase as known as synthesis phase, in which genetic material is duplicated, and an M phase in which mitosis partition the genetic material and cell divides actually. So here you can see, let's say from G1, cellular content excluding the chromosomes are duplicated. Then S phase is coming. Each of the chromosome is duplicated by the cell. So from 1 to 2 in S phase. Then in G2, cell double checks the duplicated materials for the error, making any need repairs. So it double checks everything is fine. Then mitosis happen. They divide into two parts. And then cytokinesis. They are divided into two different cells. Then they could go to cell arrest cycle also in G naught phase, where there's indefinite no division in that case. So that's how your uh, the cycle goes like this. So G1 phase is the one metabolic changes prepare the cell for division. At certain point, restriction point, cell is committed to division and moves into the S phase. In S phase, the DNA synthesis replicates. Cell is committed to division and moves into the S phase. And then DNA synthesis into the S phase. Then in S phase, DNA synthesis replicates the genetic material. Each chromosome now consists of two sister chromatids. And in G2 phase, metabolic changes assemble the cytoplasmic materials necessary for mitosis and cytokinosis. Then in M phase, nuclear division happens, that is mitosis, followed by cell division, cytokinesis, and period between G1, S, and G2, it's known as your interphase. So all this period from G1 to S to G2, this is whole period is known as your interphase. Okay, and here you have your cytokinesis, and here's your mitosis. That's how your cell cycle is divided into. Um, so then mitosis is a form of eukaryotic cell division that produces two daughter cells with the same genetic component as the parent cells. The replicated chromosomes are attached to the mitotic apparatus that aligns them and then separate the sister chromatids to produce an even partitioning of the genetic material. This separation of genetic material in mitotic nuclear division is called as karyokinosis and is followed by separation of cells in cellular division called as cytokinosis to produce two daughter cells. Mitosis is a continuous process divided into five different stages: prophase, prometaphase, metaphase, anaphase, and telophase. In adult, mitosis play a quite important role in cell replacement, wound healing, and tumor formation. So first is your prophase. So what is happening in the prophase is uh, it occupies over half of the mitosis. So half of the mitosis is done in the prophase, and a nuclear membrane breaks down to form a number of small vesicles, and nucleolus disintegrates in this case. A structure known as centrosome duplicates itself to form two daughter centrosomes that migrate to the opposites of the cells. And centrosomes organizes the production of microtubules that form the spindle fibers that constitute the mitotic spindles. Then, in prometaphase, chromosomes led by the centromere migrate to the equatorial plane in the middle line of the cell at right angles to the axis formed by centrosome. This region of mitotic spindle is known as metaphase plate. Then, in the metaphase, chromosomes align themselves along the metaphase plate of spindle apparatus. Then in anaphase, shortest stage of this is the shortest stage of mitosis. The separated chromatids are now referred in this case are daughter chromosomes. They are separated now in this case. In telophase, that is your final stage of mitosis, a reversible of many processes observed during the prophase, and nuclear membrane reforms and chromosomes group at either pole of the cells. In cytokinesis, there is a division of your cytoplasm. So here it is like this. So you have your G2 phase here. So you have already breakdown of you have sister chromatids. You have centromere here in center, sister chromatids here, and you have loosely coiled replicated chromosomes. So what is happening is there is a breakdown of nuclear membrane. Nuclear membrane is breakdown in the prophase. Your spindle fibers appears at the pole of your cell. Chromosome condense in that case. Then in the prometaphase, spindle fibers attached to the chromosomes. Chromosomes condenses. Then in the metaphase, it's aligned at 90 degrees Celsius at the center. Then in anaphase, centromeres divides. Sister chromatids move to opposite poles. In telophase, nuclear membrane reforms, chromosomes condenses, spindle fiber disappears. And cytokinesis, cytoplasm divides, parent become two daughter cells with a two identical information. So that's how your mitosis is done in this way. So bachcho, this is the most easiest and most easy way to tell if you have to study mitosis and meiosis in your life. Draw this diagram. You will never ever get failed or get low point because of this diagram. is the most easiest and understandable. Now we are done with mitosis. Now in meiosis is a form of eukaryotic cell divisions that produces haploid cells or gametes from the diploid cells uh, containing two copies of each chromosomes. The process takes the form of one DNA replication followed by two successive nuclear cellular divisions. As in mitosis, meiosis is preceded by the process of DNA replications that converts each chromosome into two sister chromatids. So in meiosis, one a special cell division reduces the cell from diploid to haploid. So in prophase one, the homologous chromosomes pair and exchange DNA to form recombinant chromosomes. Prophase is then further divided into five stages, that is leptotene, zygotene, pachytene, diplotene, and dikinesis. In leptotene, chromosome starts to condense. In zygotene, homologous chromosomes become closely associated to form pair of chromosomes consisting of four chromatids. Pachytene is the crossing over between different pairs of homologous chromosomes to form chiasmata. Diplotene is the homologous chromosome started to separate but remain attached by the chiasmata, whereas in dikinesis, homologous chromosomes continue to separate and chiasmata move to ends of the chromosomes. Then comes your pro-metaphase, same like previous one, metaphase, anaphase, telophase, cytokinesis. So this is your meiosis. Looks like this is first meiosis 1 is happening in males. So you have prophase 1, whereas chromosomes begin to condense. Then homologous chromosome pairs crossing over occurs. Then recombinant chromosomes. So first they are separated. You have two different type of chromosomes, red and blue. They got condensed. Crossing over starts to take place. Then you have recombinant chromosomes. Each have, so you can see if you go deep, so red one has blue, blue has red, red has blue, and blue has red. So like this. Then comes the metaphase. They get centered, center of the cells. The anaphase, they started to separate it by the spindle fibers. In telophase, nuclear membrane forms and telomeres lost. And then spindle fibers lost. Then cytokinesis, cells started to divide. So you have spam cell precursors in this case. So then after one part, meiosis 2 comes in which we have separate chromosomes into two chromatids. The event of meiosis are analogous to those of mitotic divisions, although the number of chromosomes involved has been halved. Meiosis generates genetic diversity through the exchange of genetic material between homologous chromosomes in the recombinant way during meiosis 1, and the random alignment of maternal and parental chromosomes in meiosis 1, random alignment of sister chromatids of meiosis 2 is happening. So here you can see that meiosis 2 in males, what is happening is prophase 2 comes. So you have again separated sperm cells. In metaphase 2 they got in align in the center. Anaphase they started to separate. In telophase the nuclear membrane forms. And then in cytokinesis you have four different sperm cells with recombinant chromatids in that case. So the red one has blue, blue has red, red one has blue, blue has red. So like this. So you have all separated very nicely. Work is done. So that's it about mitosis and meiosis as in practical. If you have been in the lab, we might have checked them in the these tests in the onion peel. So we usually done in the onion peel, or we have fixed slides. We check them during that part. So so let me just take 5 minutes break. So we meet us at 8:50 then, and then we finish the rest of the practicals in that case. So I'll also share this with you. Mitosis and meiosis. So we see us in 5 minutes at 50, 20:50. So class, I'm back. Let's start. Uh, so that was something to deal with the light microscope. I hope you have all used a light microscope in your life of your um whenever you are in the lab in your plus two in your plus one. So in your microscope, I just want to tell you which part are what. So there is a ocular, there is a tubus, there is objective revolver, and there is this objective lenses. Then there's a table, this XY table control, condenser, transmitting light source, Z control, and base sensitive. So like this, you have whole upright microscope. Um, Upright microscope looks like. This is inverted where you have a condenser on top, your objectives are down, and your camera is here. So you can it's upside down. So inverted microscope status allows the observation of living cells in culture dishes from underneath. So an environmental box can be mounted to maintain temperature and carbon dioxide concentration. So these are the various parts of microscopes. There is a detector, objective, specimens, light conditioning systems, Kohler illumination, phase ring, Wollaston prism, polarizers, filter cubes, light source. So these are the various parts of a microscope, and this is how you see from the eye. And your eyepiece is there, then it goes into your objective, then you have specimen, condenser, then you have a light source from there. So specimens type could be bulk specimens, thin specimens, live cells specimens. So bulk specimens includes kind of holder, teeth cast, scaffolds, bones. Thin specimens includes tissues, sections, cell cultures on slides where the live cell imaging of native or transfected cell layers. Fresh paraformaldehyde solutions stop cellular dynamics, preserve cells during preparations by crossing of the cellular components. Formaldehyde preparations, you have to take 37% of formaldehyde plus 10% methanol Then commercially available formaldehyde solution without methanol is unstable in time. And paraformaldehyde is a solid polymer used to prepare fresh formaldehyde solutions and use them right away for experiments. So light microscope specimens looks like this. You have a cover glass sample surrounded by embedding medium containing anti-breach agents, and you have a glass slides like this. You can see. So there are also specimen devices for inverted light microscope. You can have covered glass bottom allows the use of DIC and high resolution immersion objective, cover slip chambers, full glass bottom dish, culture dish with the glass inserts. So these are the various things you could use. You can have also 6 well plate, multi well plates, 9, 12 to check experiments, and this is done to check experiments in the full natural conditions that your cells are present, with the carbon dioxide being present in this box, and temperature, humidity, everything is maintained highly while we are doing experiments. So that was something a little bit about microscope. Now comes your last experiment: monohybrid, dihybrid and trihybrid crosses. So we have discussed them in our first lecture, but this is in the form of practical. You will have a lot of subjective details in this for you, so you can go through and have a read about it. So this is again in the practical part. Mendel's work was the first scientific explanation of inheritance was given by Mendel in 1966. He performs a series of experiments we know, and he led the foundation of genetics. Hence, he is called as father of genetics. Reason for selection of garden pea for the experiments: garden pea's annual plant completes the life cycles within 3 or 4 months. Due to this short lifespan, he was able to take 3 generations in a year. It is a small herbaceous plant that produced many seeds, and so he could grow thousands of pea plants in a small plot behind the church. It is naturally self-pollinating and was available in the form of many varieties with the contrasting characteristics, and there was no intermediate characters later on. Flowers are large enough for easy emasculations required for artificial cross and produce fertile offsprings. So these are the various characters of your dominated and recessive traits. So you can choose stem height, flower. He choose stem height, flower color, flower positions, pod shape, pod color, seed shape, seed color, with their respective contrasting traits. Tall, dwarf, violet, like this. So the reason why he was successful Mendel's experiments was that Mendel studied the inheritance of one character at a time, whereas earlier scientists had considered organism as a whole. Initially, Mendel considered the inheritance of one trait only, that is monohybrid. Then he studied two traits together, and then trihybrid also. He started with the true line, true breeding. He maintained a complete statistical record by counting on actual number of offsprings. He carried out experiments up to second and third generation. He conducted ample crosses, reciprocal crosses to eliminate the chance. He dealt with large sample size also. Monohybrid cross, a cross between two pure pattern in which inheritance pattern of only one of the contrasting characters studied, is called as monohybrid cross. It is a cross between two pure parents offspring in a single pair of contrasting characters. So first, what you have to do in this monohybrid cross explant, that you have to select your parents and obtain pure lines. So he selected pure lines of the plants, that ensuring the selected males and female plants are breeding true for selected traits. Then emasculations, dusting and raising of F1 generation. So first in the emasculation, uh, this is a process in which removal of the stamens before the formation of pollen grains. This is done by bud condition. The bud is carefully open and all stamens are removed carefully. The stigma is protected against any foreign pollen with the help of muslin bag. Dusting and raising F1 generation, then the pollen from the selected male flower are dusted on the stigma of a emasculated female flower. Like cut it from here, then put it on top. The cross-pollinated flowers were enclosed in separate bags to avoid dep- further deposition of it. Uh, The plants used as a parents are said to be present uh, to parental generation and designed for further conditions. So Punnett scare method, in the first generation you have a female dwarf, and sorry, male is dwarf, female is tall. So your cross looks like this. So you all are tall, and you have purple flower. Tall is dominant, and dwarf is recessive character. So then seeds are separated and collected in marked bottles to study characteristics of these studies immediately. But for other characters, seeds are sown to grown into next generation of the plants. So there's a reciprocal cross is done. Mendel thought F1 generation is tall because of the tallness character that he brought from the female parent, and dwarfness character is given by the male parent. To counter check if it is performed reciprocal cross, now he selected pure line plant by ensuring that selected male and female plants are breeding true for the selected traits and trait by selfing them for the three generations. He got the same results in the first case. From his he concluded that tallness is a dominant character and dwarfness is the recessive character. So he changed them. So in the earlier case, so male was dwarf, female was tall. So he changed them. He made it reciprocal, and he saw that there was still everything was same. So he then decided that tallness is the dominance character and dwarfness is the recessive character. So then F1 was self for the F2 generation. So in that case, he saw 3:1 ratio like this one. Three were tall, one was dwarf, and so pure tall is one, hybrid talls are two, and pure dwarf is one. So the genotypic ratio was 1:2:1, whereas phenotypic ratio was 3:1. Then self-breeding is done. F2 generations are then could be obtained for F3 and F4 generations. This was then checked with the other traits also, like uh, with the heights, with the color, with other types of things. And Mendel conclude that from his monohybrid cross, characters such as height of stem, color, seed are inherited separately as a discrete particles or unit. He called them as factor, determiner, or call it as a gene. Each factor exists in a contrasting alternative form, and they are factors that one of the tallness or other of the dwarfness. These two forms of genes are called alleles. And one of the factor is dominant and other factor is recessive. Only dominant factor expresses in the F1 generation. And further, he said that in an organism, inheritance of each character is controlled by each pair of factors. One of the factor contributing the male parent, and other is the female parent. Thus, higher organisms are diploid. From F2 generation, Mendel concluded that hybrid of two factors do not mix together, but they just remain together. So this is the diagrammatic representation of your monohybrid cross, tall multiplied by dwarf. Then you have a F1 generation tall, and you made a self. Then you have F2 generation to be tall, and dwarf. So that was how monohybrid cross was done. So there is also thing called, we have discussed this test cross to check which one was what was the reason behind it for the 50% tall and dwarf, whether it is the gene that is present or that one that has been repeated in this case. This we have discussed already, test cross. Uh, so there's a dihybrid cross in this. Instead of single trait, there are two traits we check. So in this case, tall and dwarf is also there, but also green pod and yellow, that is also being taken care. So here is the example of your dihybrid cross, GGTT, GGTT, and then you have F1 generation to be capital G small g, capital T small t, capital G small g, capital T small t. And then you have a gametes, GT, 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 GT. And then you made a cross, and this is how it looks like. You have a different kind of production in F2. In the trihybrid, then there will be uh, three types of characteristics will be taken care. The characters has to be considered, like seed shape, wrinkle, color of cotyledons, and flower color. So like three things are there. So they made three different crosses. having, we can see smooth yellow and violet. So it's a smooth yellow, violet, and there's a green, uh, wrinkled white. Flower color, color of cotyledon, flower color, and shape. How the seed shape is smooth or wrinkled. So you have a gametes, then you make a cross. The first F1 generation is smooth yellow violet because they are dominating. Then gametes you have like this. Then F2 generation will have 27 smooth yellow and 9 smooth green violet like this. You have different ratios being produced. So this is an example of trihybrid cross. So that's it for today. I hope you had a quite intense understanding of chi-square method, chromosomal aberrations, polyploidy, mitosis, meiosis, monohybrid, dihybrid crosses, and practical light microscopy. So that was my reason to touch you actually these topics that nobody touched them, the practical part in these workshops. So you can have a little bit about hint about these parts also. So I think I have uploaded all of them. They will be uploaded. So okay class, thank you very much. It was pleasure meeting you. And take care, stay safe, and see you tomorrow, same time. Tomorrow we will be dealing with what we will be dealing. So lack of prawn and gene expression. So these parts will be we will be dealing tomorrow on the day 5 of our workshop. Okay then, take care. Uh, See you tomorrow. You're welcome everyone. Welcome. So I will switch off.